titled Towards In-Silico CLIP-seq, Predicting Protein-RNA Interaction via Sequence-to-Signal Learning. RNA-binding proteins, or RBPs for short, are crucial actors in post-transcriptional regulation. Protocols such as iClip or eClip measure binding of a protein of interest by quantifying the number of cross-links between the protein and RNAs at each position in the transcriptome. The central idea of our method, which we named RBP-Net, is to predict the distribution of cross-link counts across an input RNA sequence. Our approach can be briefly summarized as follows. First, raw cross-links are obtained from iClip or eClip experiments. Next, transcriptome regions that are enriched with cross-link counts are selected to construct a training set. Lastly, a 1D ResNet is trained to predict the distribution of cross-link counts given an RNA sequence. A crucial aspect of RBP-Net is that it models the distribution of cross-link counts rather than the number of cross-link counts directly. As the number of measured cross-links heavily depends on the abundance of the RNA at the time of the experiment, a direct prediction of counts from the RNA sequence alone is futile. On the other hand, the distribution of counts is expected to be largely governed by RNA-dependent features. CLIP experiments are subject to technical biases, such as co-purification with other RBPs, which may have their own sequence-dependent binding specificities. Fortunately, protocols such as eClip are usually paired with a control experiment that measures the presence of unspecific cross-linking signal. RBP-Net accounts for CLIP biases by modeling the total CLIP signal as an additive mixture of two components: the control component, which can be learned directly from the control experiment, and the target component that represents the protein-specific cross-linking signal and is not directly observed. The relative contributions of the two components is controlled by a mixing coefficient pi, which is also predicted from the RNA sequence. This is demonstrated in this figure. It shows an example prediction for an RBP-Net model trained on eClip data of the QKI protein. At the top, one can see the true observed cross-linking counts for the eClip experiment in blue and for the size mesh input control in red. Below, there are the predictions for the distributions of the total, control, and target tracks respectively. As one can see, the modes of the total and target tracks correspond to the modes of the true data. On the other hand, the modes of the control track rather correspond to U-rich regions, which may stem from signal of contaminating RBPs that have a U binding motif. One can also see that the canonical QKI binding motif is recovered in the feature importance maps of the target and control experiment. As RBP-Net's architecture is purely convolutional, it can handle variable length sequences at inference time. This allows RBP-Net to perform predictions on the whole transcript in a single forward pass. This is shown here. Using single nucleotide peaks derived directly from the experimental data via pure CLIP, we can evaluate the goodness of predictions via an area under ROC score. This we do by simply treating nucleotides that fall within peak regions as positives and the rest as negatives. Here we show the area under ROC scores for RBP-Net on the y-axis, as well as for a classification-based method, DeepRipe, on the x-axis. As we can see, for each of the 70 evaluated eClip experiments, RBP-Net outperforms classification-based methods on all of them. In addition to prediction, RBP-Net may also be used for variant impact scoring. This is done by leveraging the fact that in-silico perturbation uses a change in the distribution predicted from the sequence. So here we can see that for the reference distribution, we have a predicted distribution, or for the reference sequence, we have a predicted distribution in blue with two very sharp modes, while if we induce a C2A mutation, these two modes drop significantly and the probability mass that's predicted gets reallocated to different positions. We can quantify this the change in distribution between the reference and the alternative sequence as the KL divergence between the two distributions. For this example, we can also see that the impact of the variant is manifested in the feature attribution maps. So a highly positive feature importance / attribution becomes a highly negative one. Lastly, ABPNet can also be used to construct consensus motifs which reflect an ABP's sequence affinity. This is done by first computing predictions around thousands of peak regions for given ECLIP experiments, and then afterwards computing feature importance scores for those predictions. From these feature importance scores, we then extract the 5-mer that has the highest importance for each peak region, and 5-mers are then aligned greedily, starting with the 5-mer that has the highest importance in order to build consensus motifs. Some example consensus motifs for proteins ABFOX2, TIA1, QKI, AQR, U2AF2, and CSTF2 are shown here. To summarize, ABPNet is a method to predict protein-RNA interactions at nucleotide resolution. It enables prediction of variable length sequences and even across whole transcripts. ABPNet's feature importance scores can be used to build consensus motifs that reflect an ABP binding specificity, while variant impact scores can be computed via in-silico perturbation. If you're curious for more, either meet me at the conference or scan this QR code here, which links to our preprint. Thank you very much for listening to this presentation.